Hi, everyone, and thank you for joining us today. So today we will talk about the data and why is it the engine behind AI-driven drug discovery. But first, a short introduction. CAS is a division of the American Chemical Society. This is a non-for-profit organization that is devoted to accelerate science. At CAS, we believe that data is valuable only when it's transformed into insights. And this is why our experts, which are all scientists, are harmonizing data, collecting data, harmonizing data. They organize it and make the right connections. In order to enhance the value of the data, our experts also use our ontology system, lexicon, thesauri, in order to increase the value of the data to make the right context and connections. On top of this data foundation, which we call CAS data collection, we have our technologies like the tools you are using, SciFinder, IP Finder, BioFinder, which is our new drug discovery tool. These technologies are actually the gate into our data. On top of this, let's say, pyramid, we also have our AI models, our AI agents that supports AI related insights. The global AI in drug discovery market size was valued at about $1.2 billion in 2023, about $2.5 billion in 2026, and is projected to reach about $13.6 billion by the end of 2033, rising at a CAGR of about 25% between 2026 to 2033. The rising demand for cost-effective drug development, the increasing number of clinical trials for drug repurposing, and the increasing rate of chronic diseases are significant factors contributing to this market growth. Our CAS experts performed a quantitative analysis of our CAS content collection, which is the largest human-curated repository of scientific information in the world. This image represents about 74,000 patent families. All are AI-related publications, patent publications, that were published in the last 20 years. As you can see from this image, numerous fields have been impacted by AI. But biochemistry, the green curve over here, is really overshadowing all other disciplines with a very strong exponential-like increase. AI has transformed biomedical research by solving problems that were previously either too complex or too expensive or too slow to address experimentally. This figure also highlights the intense commercial interest and investments in life sciences. We know that AI in drug discovery and in life science in general is growing rapidly. But where is it challenged? Why is it not going faster than it is? We believe that the main challenge is the data. We know that data is only as good as its data. And this is exactly the challenge that CAS addresses over the last century and more. CAS transforms scientific data into high-quality, AI-ready resource that many pharma companies already rely on to strengthen their AI models. For example, what we do is basically to extract data from many sources. We know that drug discovery data is extremely complex, it's fragmented, it's siloed, because it's coming from different sources, different databases. We make it, we harmonize it and make it become a single consistent foundation. For example, we also use authority constructs. When you look at this protein, for example, you can see that there are many names or IDs that exist across publications, across different databases. CAS brings all these variants into a one single authoritative record, enabling consistent, reliable use of standard identifiers across discovery workflows. For that purpose, we use human expertise together with AI-assisted expertise. We do think that human expertise are highly important for this type of complex sets of data. Human in the loop is necessary. Human curation creates high-quality training data, reducing bias and improving AI model reliability. Today, I want to show you two examples of how better data will lead to better results. We have two examples. One is predictive models for drug target activity, and the other is life sciences knowledge graphs. Let's start with predictive models. In BioFinder, which is our new drug discovery tool, we use a set of 5 models to predict the P activity value, which is basically the drug target activity prediction constant. Here are 2 examples of a ligand and the target and the experimental P activity. Now we use the set of 5 models to predict those models, and we came up with these numbers, which are very far from what they should be. And no, I didn't mix the numbers. This is really how it should be. So what we did next is we took our harmonized bioactivity data, our AI-ready bioactivity data that our scientists curated. We retrained those 5 models again. And here's what we got from the retrained model. As you can see here, much closer to what it should be. And this is actually showing a reduction of 56% in the difference between predicted and experimental, also a 23% reduction of the standard deviation. So even though we are using commercial models, you do need to have the right data. The next example relates to knowledge graphs for life sciences. Knowledge graphs or building knowledge graphs is, especially for drug discovery, will always start with bringing together all types of data points from many disconnected sources, from the literature, from patents, databases, experimental results, compound libraries, omics libraries, bioactivity data, clinical information, really a lot of sources. These sources almost never speak the same language. And so the first step in building a knowledge graph would be to harmonize terminologies, to harmonize structures, identifiers, and relationships. Life sciences knowledge graphs are powerful because biology is fundamentally relational, right? So we know that proteins interact, drugs bind targets, disease-involved pathways, and so on. Representing all this as a knowledge graph really allows AI and analytics to predict the drug target interactions, to identify repurposing opportunities, to find disease mechanisms, and a lot more. Of course, again, we use not only human curation, but also machine-assisted curation. And this is more or less how it looks like. We have the data, all types of domains that are connected in a way that eventually will help our AI system to make smart predictions. CAS really excels in that, in creating those connections, right? First of all, this is what we do, right? We harmonize, we standardize, and we connect across different sources and databases for many years. We know how to do that. Second of all, we have the correct high-quality data and architecture for this data. We use the right taxonomies, ontologies. The ontology system is highly important. This is used, of course, to create consistency. And of course, we use this combination of human and machine curation so that we have our domain experts that will validate and enrich relationships, while AI will speed up and improve the extraction. And this really results in a high fidelity, trustworthy graph that really supports advanced analytics and AI-driven discovery and decision. Coming back to this curve that we saw earlier, but now I want to focus on the way it breaks down the growth by component, software versus services. And what does it mean? Software growth means any increase in spending on AI products that the scientists are actively using, like platforms, licenses, subscriptions, like, for example, the AI agent Newton on SciFinder. Services growth represents the rising revenue from outsourced sources, projects to specialized companies on behalf of the drug developers. This breakdown to software versus services also presents how CAS delivers value across the discovery landscape, because we have our CAS tools. We blend deep human expertise that are embedded in our taxonomies, ontologies, the curated data, together with our AI algorithms, which are CAS Newton. If you haven't tried it, this is a good opportunity to try it. And we have our customized services, in which we provide specialized project-specific support, more customized projects, in which our data scientists, which are, of course, also scientists, are helping customers, for example, to create the correct data curation from internal or external data sources, create AI-ready sets, run pipeline development, ontology design, knowledge graph enrichment, as we saw before, and really a wide range of any data-driven capabilities that we can offer. And here's how you can actually partner with us. You all know our solutions, at least some of them. We have SciFinder, we have BioFinder, we have IP Finder, we have our AI agent, CAS Newton, on top of all of those. We also have our workflow integration. We recently launched CAS Connections, which allow a connection through an MCP server to our tools. We have the API integration, agent-to-agent interactions. We also have our consulting options, custom data solutions, and of course, data management tools. This is our CAS Intelligence Hub. The CAS Intelligence Hub is relatively new. It actually allows you to take your internal data, the same process as we take our data through to make it AI ready. Same process of human curation and harmonization of the complex scientific data that you have. We can make sure that it's done according to our taxonomies, ontology system, and so on. This is our what we call CAS Intelligence Hub. It's fundamentally new, and it really opens new opportunities for you. In a bit more details, A common data environment brings together all your scattered systems where your scientific data currently lives. It's fragmented, it's siloed, of course. While on the other side, you can see the CAS infrastructure, which shows how data can actually be consolidated and managed more effectively. We can support you and connect at any stage of this consolidation process. The more refined and harmonized your data becomes, the stronger and more reliable your AI outputs will then be effective. And this can be connected, internal, external, and so on. To summarize, data is the driving force of all AI solutions. High quality and well-structured data is really essential for a reliable AI workflow for drug discovery. A strong data architecture is very, very important. We really need to have the right data architecture, structure, connections for these complex life sciences or specifically drug discovery data. We do believe that a human in the loop is a must to be able to support this complex scientific information to become accurate, connected, and usable correctly. Altogether, those three elements will create a trustworthy foundation for advanced analytics and AI-driven discovery. With that, I welcome you to contact us to discover how CAS data solutions can power your AI initiatives or consult with us. And I'm happy to answer any question.
Thanks so much, Ayelet, for the great presentation. I'm now going to hand things over to Julia and also back to Ayelet to go through any questions that we received during the presentation.
Thank you, Christy. Thank you, Ayelet. Yes, we have some questions. Let's see. That 56% accuracy improvement that you presented, Ayelet, is that possible to be done with the audience members' proprietary targets and compounds?
You mean, I guess one means these and the question, can you repeat the end of the question, please?
Can that be done with our proprietary targets and compounds?
Yes, exactly. Yes. When we talk about AI models, basically the more accurate data that we have and more comprehensive data that we have, we will gain more accuracy in prediction. And of course, if we take internal data, we make the right connections, we harmonize it correctly, and make the right context and connection between the data points, we can then structure your data, your legacy data, let's say, in the same way where we structure our data. And therefore, combining all of those will always increase those numbers, basically. The prediction will always be more accurate.
I guess along that line, the question is, we have decades of legacy lab data in a lot of different formats. Can that be made useful? Can we use that?
Yes, I think this is exactly, it goes straight to the Intel Hub, where the legacy data is probably, and it depends on the size and how long the company is already functioning and working, because you might have like years back, different sites scattered around the world, but not only. And each group or each department or each site will have their own data sources. And this is why we present the infrastructure like that, because all those sources are kind of connected, but not really, because they don't speak the same language. You cannot search all of them all together. And what we offer here with the Intelligence Hub is that we take care of this silo data, this inconsistency that you probably have in your data infrastructure, and we can make it harmonized and AI-ready data. It will actually look like the other side here, which is just one block, one database, one data source. All the data architecturally looks actually the same, and so you can have them the right predictions, and you can use it not even only for AI initiatives, but also for everything else. You can really search your data in a way that will actually help you. We see that many digitization projects fail because the data is not structured correctly. And so this is where we can assist.
There are some questions about AI versus the human expert. Can you talk about where those two play when it comes to the harmonization of data?
Yes. So we see that there are many efforts in the last few years trying to, let's say, curate data automatically using AI models or using all types of resources. We see a good result, but not in life sciences, because it's very complex. You have, if you look at chemistry and you have a structure, it's a 3D structure. It's very hard to understand how to curate it. Also, when it comes to pathways, to formulations. So this data is too complex for AI or machine-assisted curation by its own. We hear a lot of things going on, but nothing is yet there. It may be in the future, but not yet. And so we must have a human in the loop. It's like we always hear we must have lab in the loop, because we can't only trust the predictions of AI. We have to work in a wet lab to check those things. It's more or less like that. AI is a good assistance, but it cannot replace human curation.
Thank you. Another question is about experimental data from patent literature and how is it used in CAS data? How reliable is it when it comes to CAS predictive analytics?
Depends what we consider analytics data. Is it like analytical chemistry methods that are? I will assume it is. Yeah.
I'm not sure. Yeah. How experimental data from patent literature is used in CAS data collection and how reliable it is in CAS predictive analytics. I interpreted it as
Okay.
CAS as, okay, you got it.
Yeah. Okay. So yes, our CAS experts are going through the entire patent, even if it's 500 in, I don't know, Chinese or German or English. They read the whole thing. So each expert, and we have many of them, which all have PhD in specific scientific domains, they all together speak 50 languages. And each one of them will receive the publication that is of his knowledge. So they will get the patent, they will read through everything, including all the attachments and notes and experimental and everything. And they will extract this information. So yes, we do use this data. For example, we have the synthetic routes, we have experimental routes, we have formulation processes, and so on. And so this experimental data, together with properties, of course, and so on, is very accurate because we do use machine-assisted technologies. But we do have, so the last, let's say, layer is a human that makes sure that we didn't miss anything and we didn't, I mean, there will always be mistakes, but I think it's as much as accurate as it can be.
Thank
If I
you.
understood the question correctly.
I think so. You talked about knowledge graphs. Knowledge graphs, is it possible to build them that includes that proprietary data, that customer's proprietary data alongside CAS content?
Yes, of course. So we have some several different projects. For some projects, we do help the customer build their semantic layer so they can kind of work internally on their data. However, there are projects in which we get involved much more. And this is where we use internal data together with external data to maximize the way that the graph is built. And so, yes, we can do whatever is decided in the company. We can get pretty much very involved or just a little bit. But yeah, of course, it can be a combination. It can be only external data. It can be only internal data. Absolutely.